Okay, our last case study we'll be looking at is another case of binding of ligands to proteins in a way that, in this case, helps us move. So here we're going to be using the muscles as an example of protein interaction. Now, muscles are made up of these large bundles within very long cells. In fact, muscle cells are very elongated. They're multinucleated, meaning there's multiple nuclei within one cell. And they have these very, 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 very long bundles of muscle fibers. So these are a type of protein that are fibrous in nature that make up these bundles. The main proteins that make up these bundles, one of them is called myofibrils, which make up what we call the thick filaments of the muscle. This protein is called myosin, which makes up these, again, myofibrils. Myosin makes up fibers to make myofibrils. These are made of supercoiled alpha helices. And you can see an example of this here. Here's myosin that's then coiled into these, again, what we call supercoiled alpha helices. Now, myofibrils also contain the thin filament protein, which is the protein actin. And see, this is also a coiled type of protein made up of subunits, actin subunits, to form this whole actin filament. The myosin and the actin act together to help in the stretching and movement of muscles. So myosin thick filaments, which we discussed, actually slide along the actin thin filaments. Here's what that looks like in a little bit more detail. You have these, again, the thick and the thin filaments. So the myosin thick filaments in the center here actually move along these thin filaments of actin, and they kind of crawl along. You can see if the thick filaments start crawling in either direction, they will essentially pull the actin in, contracting the actin around it, and that is how we have muscle contraction. So you can go from a relaxed state to the contracted state, and that's the very essential way that muscles move, is going from relaxed to contracted state. But how, on a biochemical level, does this happen? It's very easy to say, oh, the myosin crawls along actin, but how is it actually crawling? It's happening through chemical reactions. What's happening is that ATP is actually being utilized to bind or unbind, causing conformational changes. And that conformational change in the protein will change its function. This is a very common way in which many of the proteins which in biological organisms act. They'll use ATP to cause conformational changes. And especially this occurs in motor proteins. So control and movement of cells and organelles within cells is very often controlled by the use of ATP. For muscle cells in particular, muscle contraction occurs through a series of these conformational changes. And again, it's due to protein structure binding, hydrolysis, and then release of ATP. So you can see a little bit more of a close-up here. Essentially what's occurring is normally you have a part of the myosin, which we call the myosin head, which is interacting with another portion of the actin. So these are two proteins that have some interaction between them, some non-covalent interaction. When ATP binds to myosin, you can see it causes it to stop interacting with the actin filament. So this is a type of ligand binding in which we're causing a change in the structure, and that change results in myosin and actin no longer interacting. But as the bound ATP is hydrolyzed, so through a chemical reaction in which ATP is broken down into ADP, the ADP and the remaining phosphate that is there then associate the myosin head with the next portion, next actin along the actin fiber. This allows that phosphate to release, and now you just have ADP that is bound to the myosin portion. When the final release occurs of the phosphate, it actually triggers what we call a power stroke. And again, this is just a conformational change that moves the head to that next actin molecule. And also, in tandem, the myosin filament opens to another potential ATP binding, releasing its ADP. And the process of binding an ATP and then this hydrolysis and release and movement that happens again and again and again. And this is all because of conformational changes that are just occurring from ligand binding and ligand release, causes that myosin to essentially one by one crawl down the end of an actin fiber. And as it does that, it contracts, right? So as it crawls and crawls and crawls, it contracts the whole of that cell, it contracts the whole of that muscle fiber, and contracts essentially to form of movement in an organism. This is how we move our arm or move anything that requires a muscle. It's multiple use of ATP and ATP hydrolysis to slowly, well, very quickly actually move that myosin head down the actin fiber. This, of course, occurs at such speed, such quick speed, for us to be able to move our muscles very quickly. Now, there is also another type of regulation that's occurring in muscle cells, and it's occurring through ligand binding. When I just discussed use of ATP hydrolysis to contract a muscle, if that was always available to our muscles, we essentially would always be in a contracted state. If you always had your muscles utilizing ATP and moving that myosin, your muscle would always be contracted. But the organism needs an ability to turn this on and off. We need the ability to essentially utilize the conformational change of ATP and contract when needed, but then go back to a relaxed state and not be utilizing ATP also when needed. The way this is done is through calcium binding. The availability of those myosin binding sites to the actin is regulated by another protein, this troponin and tropomyosin. So they're another protein that wraps around the actin filament. The tropin and tropomyosin protein are going to be controlled by binding of calcium. Essentially, what happens is that when your brain releases calcium into your muscles, that's the trigger to move your muscles. So, a nerve impulse that travels to the muscles will release calcium. Calcium can then bind to this tropomyosin. It makes a conformational change in tropomyosin, which moves tropomyosin away, exposing the myosin and actin binding and allowing that previous actin-myosin cycle to occur. So then you can allow actin and myosin to interact, utilizing ATP and this movement to occur to make contraction. So muscle cells are rife with types of conformational changes that allow us to use them in a way that's just occurring through ligand interactions with proteins and causing those conformational changes. Now, I know some of these types of conformational changes are hard to just see in slides like this, and I recommend that you utilize the link I've provided in Blackboard, and it actually shows you a short video of how binding is occurring in muscles. So first, calcium binding, which exposes the actin by changing tropomyosin, and then the actin and myosin interaction, which utilizes ATP binding to have conformational changes to cause contraction in muscles. Okay, so that is it for Chapter 5 summary. Remember, we generally discussed protein function in a variety of ways, but all of these relate to how ligand binding affects protein functions. We talked about a way to analyze binding data and understanding a Hill plot and the Hill equation. And we looked at different types of case studies of protein function and protein binding. So we looked at myoglobin and how it stores oxygen. We looked at hemoglobin and how cooperativity can affect ligand binding in the example of hemoglobin. We looked at antibodies and how changes to the actual structure of the exterior of a protein can cause it to recognize and bind different ligands, which is how antibodies work. And then we looked at how structural changes, because of antibody binding and conformational changes, can help with something like motor function in muscles.